Hello everyone. So we have started discussing the causes of antimicrobial resistance and we have looked at the very first factor, which was selective pressure. Now we are moving to mutation. Okay. I can understand that for those people who are from, non-biology background, this may sound a little technical, but it's not very difficult to understand. All characteristics that living organisms exhibit is because of the genes that they possess. Genes are fragments of our DNA. DNA is our genetic material. So that genetic material is very long. If you look at the fragments of this genetic material, we refer to them as genes. Every gene may be responsible for giving you a specific characteristic. So at times, these genes, which are like recipes for the characteristics that you possess, may undergo some small changes. And it's like cooking, that if there is some change in the recipe, there is going to be some change in the flavor as well. So something similar happens with mutation, that if there is a slight change in the gene, what the characteristic was coded by this gene may slightly change. So some bacteria undergo such changes, and these changes that happen in their gene give them the ability to resist antimicrobial agents and survive in the presence of antimicrobial agents. So mutation, that is changes in the DNA, could be responsible for the bacteria becoming resistant to an antibiotic. So in a population of bacterial cells, all will not get this mutation at the same time. One may get it, but that one cell, when keeps on multiplying, it is keep on passing that character to the future generations. And all those future generations who have that mutation will show the resistance. This is what I was speaking in the last few videos, that nature is so impartial, it's not biased. It tries to protect each and every creation that it has made by these kind of techniques like selective pressure or mutation. It's giving an opportunity to every single creation of its to survive, to exist in the environment. Then comes gene transfer. This is the worst of all, and this is so much important to understand how antimicrobial resistance spreads. We said in the last 2 points that an organism may have resistance or it may gain resistance due to mutations, but it also has the ability to pass this characteristic to its counterparts. So imagine if I am a bacteria and if I gain resistance, I also have the ability to pass on this characteristic of antimicrobial resistance to my counterparts, to other bacteria. And this is how resistance will spread from one organism to the other. So if there is a resistant bacteria and if there is a non-resistant bacteria, as it is shown in the image towards the extreme left, if you get them in contact, they may end up exchanging some of their genes, which are part of their genetic material, and the resistant strain may just end up giving the resistance characteristic to the ones which did not have resistance. And that's how now everyone in the population starts gaining resistance. This is risky. So even if one bacterial cell gains resistance and if it starts passing on these characteristics to the other counterparts in the population, that's it, we are destroyed. There is going to be spread of antimicrobial resistance to such a great extent that we can't even imagine of. So how does this gene transfer exactly take place? So again, we are going to speak about some technicalities, but I'm trying to make them as simple as possible yet again. So there are three major processes. transformation, conjugation, and transduction. This is the mechanism by which gene transfer takes place in microorganisms. Let's start first with transformation. So let us say that I am a bacterial cell who does not have resistance. I do not have resistance. And I have other bacterial cells around me who have resistance with them. And due to some condition, some of them die. And as they die, they are going to release their genetic material outside. Because once the cell is dead, the cell breaks open, all its contents start leaking out. So even its genetic material comes out. I am still alive. Okay. So now as they start leaking out their genetic material, I can pick up some fragments out, from outside, and I will hold it with me. So now if I pick up the genes for resistance from their genetic material, now I am transformed. Because I did not have resistance initially, I ended up picking up a gene from outside which coded for resistance. So now I have transformed from a non-resistant bacteria to a resistant bacteria. So hence the term transformation, wherein genes are picked up from the environment, from outside. Next comes conjugation. So let us say that there are two bacterial cells, one which has resistance, another which lacks resistance, and if they are in close proximity, as you can see in the image also at the center, the resistant organism will insert an appendage into the non-resistant one and will pass on its genetic material from that tube, so that now the gene is received by the non-resistant one. So this is actually coming in close contact with each other, forming a bridge and exchanging the genetic material. So the resistant strain can pass a copy of resistance to the non-resistant one, and now even this bacteria has turned resistant. So organisms, these cells can have multiple copies which they can share with each other by this process of conjugation, where they conjugate. The live cells come close in contact, they have a bridge formed between them, and they are exchanging the genetic material. So that's conjugation. We also discussed, in the very first week, that there are viruses which can infect bacteria, like bacteriophages. So what do these phages do? Let us say that a phage infects me. I am a bacteria. Let us assume I am a bacteria. So the phage infects me. Okay. So when it infects me, it is going to combine its DNA with my DNA. So both of the DNA has combined now with each other. Okay. Now when this phage multiplies and now wants to leave my body and wants to infect other bacteria, it will try to extract its genetic material which had combined with my genetic material. Both of our genetic material were in combined form. Now as it wants to leave my body, it wants to separate its genetic material from mine. Now while it does this, it ends up taking some part of my genetic material also along with it. Because the cut mechanism is not perfect. It doesn't understand where exactly it has to cut. So it may end up taking some part of, my genetic material, who is right now a bacteria, along with it. And if that part that it has taken with it codes for resistance. So let us assume now this phage goes and infects another bacteria which is not having resistance. And as this phage infects it, it has got the resistance gene which came from me, and now it is being passed on to that other bacteria. So phages, viruses, are transferring resistance gene from one bacteria to the other bacteria. That's transduction. So I have tried to simplify to as much limit, I could have done it. If you have not understood this, try to rewind the video and hear it again. It makes sense. Okay, it will make sense, I'm sure. It takes some time for non-science background people, but it will make sense to you. So we have discussed all technical aspects, and now we will move to the other aspects which are responsible for causing antimicrobial resistance. So stay tuned.